But how can we use what we know about genes and the evolution of these developmental genes to infer about the evolutionary history of a trait? An important example in animals, where we used to think that segmentation, there are many animals that are segmented, and you probably heard that arthropods are segmented, annelids are segmented, and us chordates, we are segmented. And for a long time, we used to think that segmentation, which we see again in these groups, the annelids, the arthropods, and chordates, they all show segmentation. We used to think that this trait had evolved in a single clade, or that this was a homologous trait, that it was shared by the ancestor of these three groups. But now that we understand the evolution of the genes involved, we know that this is not the case. So segmentation is regulated by Hox genes, and these genes are present in the ancestor of all these organisms, but those genes duplicated in 3 different times. So these are 3 separate gene duplication events that resulted in segmentation in each of these 3 groups. So this shows that segmentation is not a homologous trait in these 3 groups as we used to think, but is actually an analogous trait, so something that evolved independently in each of these 3 groups. Likewise, understanding the genes that regulate development of different structures has helped us understand the origin of those structures. For a long time, and when I was in college, for example, we used to think that eyes had evolved independently in different animal groups. So we thought that the eyes, for instance, of mollusk, like the squid here, had evolved independently from the eyes of arthropods and the eyes of vertebrates. But now, looking at the genes that regulate the expression of eyes in those groups, we now know that actually eyes evolved in the ancestor of all animals, and different changes in the eye have been modifications to that original eye structure. And we know this because the same gene that initiates eye development is the same gene for all the species. This is an example of the PAX6 gene regulating the expression of eye development in multiple species. So we've seen how it regulates it in invertebrates and also invertebrates. And this is an example of fish species. This is a fish that lives in regular waters. It lives in the surface where it needs its eyes in order to find predators and find its food. And the one here below is a species that lives in a cave. And since they live in a dark environment, there is no need to have eyes. And they've actually lost the PAX6 gene, and as a result, they've lost the expression of their eyes. They do not have eyes. So this is an example of how losing one gene, the function of one gene, this PAX6 gene, which is the main transcription factor in the formation of eyes, results in the complete loss of that structure. And we have studies that have addressed this question of whether the same transcription factor regulates this eye structure, the formation of eyes in different species. For example, in this experiment, we took a fruit fly, and here you can see the eye of the fruit fly, and on the leg, a transcription factor for eye development in mouse was inserted in this fruit fly. So this part here, we zoom in, we have this image. This is where the transcription factor for mouse was inserted, and this is the same transcription factor that regulates eye development. And when you put it in the fruit fly, even though it's the gene from a mouse, it still promotes the formation of eyes in this fruit fly. And the eye that develops is a fruit fly eye. And we can see it is a fruit fly eye because fruit flies have compound eyes, while mice have the same eye as vertebrates we have. So you can see here the multiple elements that form a compound eye, showing that even though it's a mouse gene, it still promotes the formation of a fruit fly eye. The fruit fly eye example is similar to the genes that regulate limb development in tetrapods that we've mentioned before. So we said how this transcription factor TVX5 regulates limb development in ancestral tetrapods, it regulates it in humans, and it also regulates it in birds. Although different genes are activated in these different species, the same transcription factor is responsible for activating those genes involved in limb development. And the result, even though it's the same transcription factor, it would activate different genes on each species. So if you see here, it's activating this, in humans it activates this pink gene and purple gene and the brown gene, while in birds it activates the pink, the yellow and the blue gene. So we can find examples of genes that control development in multiple organisms. One important example are these homeotic genes or Hox genes. These Hox genes are present in organisms as different as Drosophila or a fruit fly, and in vertebrates, including mice and ourselves. And these genes, one interesting thing to note is that they're organized. You see here, they're color-coded by the region in the embryo in which they are expressed. And you can see that the genes that are most anterior in the chromosome are also the genes that are expressed in the anterior part of the embryo. And the same is true for vertebrates, where the most anterior genes in the chromosome are also those that are expressed in the most anterior part of the embryo. So this shows the homology between these genes and the structures that they are regulating. But another interesting thing is how these are the same genes from the same ancestry, and how they are controlling same regions or similar regions in the body. Yet, if you look at where that results, are very different. So here, this gene in the fruit fly, the gene in purple, is regulating the formation of the head in the fruit fly, but that same gene here in the mice in the purple is regulating the formation of part of the cervical column. So these are same genes but are regulating very different structures. And this shows us that in evolution, evolution is used in the same toolbox but to produce very different results. One way to look at this analogy is like seeing these developmental genes are acting like orchestra conductors. So they are deciding which genes play for how long in which particular location, but what song the genes are playing, or rather what instrument they're playing, depends on the organism they're in. So if you have an orchestra that only has drums, then it is going to play the same song, but it's going to use drums. So to bring that back to animals, the same gene that regulates development of mice, if you put it in a fruit fly, it is going to develop fruit fly eyes. So it is an orchestral conductor that plays the song of eyes, and if you put it in a mouse orchestra, it is going to play a mouse eye, and if you put it in a fruit fly orchestra, then it's going to play a fruit fly eye, to say it in a way.